Okay, it's 12:01, so I figure I'll start this introduction, and I'm sure a few more people will trickle in as we talk. But welcome. This webinar is presented by University of Maryland Extension and the Woodland Stewardship Education Program. A few housekeeping items, and you might have noticed when you entered that this webinar is being recorded, and it will be made available on our website at extension.umd.edu/woodland. And the benefits of you registering for the webinar is you get added to a quarterly newsletter and a notification of upcoming events. And if you'd like to opt out of those items, you can email Pam Thomas at pthomas@umd.edu with a message, no events. If you have any questions or comments during the presentation, at the bottom right of your screen, there is, in the control panel, there's a little button that says chat at the bottom of it, and you can click that and add any questions or comments you might have. And also feel free to introduce yourself. I see Norm Pattison just introduced himself, so thanks for doing that, Norman. Everybody else is welcome to do that as well. And we'll have a brief survey at the end. When you log out, you should be automatically taken to a survey where you can give us feedback and things you'd like to hear about in the future. So today's webinar, I'm really thrilled to have Dr. Jennifer Mullinax presenting on mitigating the wildlife and human risk of contact with ticks in Maryland. And that picture really makes me kind of squirm. So hopefully, I'm sure it'll engage a lot of us as we think about these important aspects of disease and Lyme disease with ticks in Maryland. Next month's webinar, looking forward to that one as well. It's going to be on June 15th. It'll be presented by Dr. Deborah Landau with the Nature Conservancy, and she's going to speak to us about fire history and ecology of Maryland, and with a tie-in to what that fire history means for wildlife. So I've heard great things about Dr. Landau's presentation, so I encourage everybody to mark your calendars for that. We have a lineup for the rest of the year covering all these different topics. We'll be covering weeds on July 20th, sustainable forestry in August 17th, forest stand improvement for wildlife with Dr. Craig Harper in Tennessee, and bats in October in honor of Maryland's Halloween. And finally, we'll have a forest health and pest and disease update on November 6th session. So with that, I want to introduce today's speaker, Dr. Jennifer Mullinax. She is an assistant professor at the University of Maryland's Environmental Science ENSC, Environmental Science and Technology Department. She is a research ecologist, and she describes herself as a professional asker of questions. Her work is motivated by a sense of community, curiosity, and open-mindedness. Jennifer is interested in the nature of the patterns of animal behaviors and movements. By combining natural history, quantitative analyses, and modeling, she attempts to define the interactions between humans, animals, and the environment. She enjoys using these methods to find applied solutions to specific wildlife management issues. With that, I will continue, and I believe we will have a poll pop up where people can share where they are from in the state. Taylor is going to pop that up for everybody. And you can say what part of the state you're in, what county. We have people from all over, a lot. The biggest group is 3 from Central Maryland, but we have a good distribution across the whole state. One person's from outside of Maryland even joining us today. So great to see that broad participation across the state. We'll close this out now and go to our final slide and let you know if you have any questions about this. I want to welcome everybody and encourage you to ask questions on the chat and engage with us. We'll have question and answer at the end. If you have any questions in the future, speaker topics, or anything else, feel free to email me at lucmac@umd.edu. And with that, I am going to stop my sharing and allow Dr. Mullinax to share her screen.
Okay. Welcome, everyone. It's great to be here today. Let's see if technology cooperates. Okay, so I'm here at the main campus at UMD in the College of Agriculture and Natural Resources, as Luke said, in environmental science and technology. By training, I'm a wildlife biologist, wildlife ecologist. And since my school days long ago, I've found myself really interested in how animals move. And I came to Maryland, and I actually was just a lecturer, and then shifted into more of a research component in 2017. Not just a lecturer, that's actually probably one of the most important things that we do here. But the research component of my lab didn't start until 2017. And it's taken some interesting directions. I study many different topics, some related to conservation and fragmentation, but a huge section of my lab is now dedicated towards zoonotic diseases in the environment and the specific roles that wildlife play. Today, I'm going to be talking to you about ticks, specifically in Maryland, specifically hard-bodied ticks. So that's the primary ticks that we have are the dog tick, the black-legged tick, and the Lone Star tick. Most of the stuff I'll be talking to you about today have to do with black-legged tick because it's directly related to Lyme disease. Although the things that we're going to be talking about actually extend to several other tick-borne diseases. We have just wrapped up a big project in Howard County, Maryland, which we'll talk about, and we're starting a huge project in Montgomery County, Maryland, and some pretty interesting things that we're finding. Let me, I'm going to hide this floating meeting control for now. Okay. So I'm sure that most of you are familiar with Lyme disease because we are at the very southern end of the high incident area. What you're looking at right now is a 2019 map of confirmed CDC cases of Lyme disease. And so, you know, as is probably common knowledge to most of you, we are smack dab in it. And in terms of us being concerned with this, what we know, okay, I'm seeing myself, I don't know if we're still, okay, you're still seeing that, okay.
No, we don't see your screen.
Are you seeing
Yeah,
this?
I'm not sure what
slide?
happened.
I'm sorry.
No,
The share seems to stop.
Okay, let's see.
Coming back now.
So what we see is that the, I'm gonna hide these controls again. What we see is that with changes in either yearly actual climate events and long-term climate events, we see the tick distribution actually spreading, and that's causing concern for those of us who are paying attention to the zoonotic world. So in general, we think of tick habitat and we think of this. We think of tents and woods, being outside, hiking, maybe people who have professions that are going to lend themselves to more rural areas, in areas of scrubby vegetation. That's pretty much the norm. What's interesting is that when you start getting into the actual biology of ticks, remember, I'm not an entomologist, so I had to learn all of this on the back end. It's really complicated. It's really complicated because they go through this 3 cycles of the ticks are hatch, you know, the eggs are laid and the eggs hatch into a larva. But at that stage, which is good for us from a standpoint of disease, they aren't carrying any kind of bacteria. The bacterium Borrelia Lyme that causes Lyme doesn't pass from the mother to the eggs that she lays. And so the eggs are fresh. They molt into what's called a larva, and then it has a blood meal, usually on a small mammal. They actually end up picking up these bacteria from those wildlife species that they're feeding on. hence the wildlife ecologist being a part of this conversation. Then they go through this complicated set of year one, wherein they live a year, they've had this blood meal, they live a year, and they molt into a nymph, which is kind of this middle stage. And those are the ones when people, they call them seed ticks, and they're like covered in hundreds. You can step into a nest. It's often the nymphal stage, and they're very small and much harder for us to see on our body. These also have a blood meal. Now these species at this stage can feed on small mammals all the way up to humans. And so we have to be concerned with it. And then they have that second blood meal and they molt into an adult. They have another blood meal right before they lay eggs. And so this is a pretty long cycle. So if you're going to address any kind of tick issue, you've already got a built-in 3 years that you have to actually address. From my standpoint, in terms of me trying to contribute to Lyme disease, I'm most interested in these mammal hosts, because lo and behold, they are the culprit in terms of not only keeping ticks in the environment, but actually giving ticks the diseases that they bite us, and then there's a blood exchange and we end up getting from them. And so they're the vital component in terms of how do we potentially stop this from maybe an integrated pest management perspective. One step further, it gets, when you start getting into how this works with hosts, it gets incredibly complicated, because not only are you interested in like the small mammal, the mouse, but then you have to think about, you're going to have good years and bad years. You have a big acorn crop, suddenly you've got a huge mouse population that has major implications for ticks and Lyme. Are you missing predators in the system that could help, maybe not depress the small mammal population, but potentially cap it? And then how is your deer population? Deer originally in Lyme research got a really bad rap. Lo and behold, they're not very good at giving ticks Borrelia, Lyme. What they are good at is supporting a lot of ticks so that the general tick population stays high. And so it's this very complicated interweaving of these different communities, from vegetation to small mammals to predator-prey dynamics to these large ungulates. And then you've got these different stages. And then you add on top of that, throughout the year, you've got different timing of when these ticks are out there, what stage is out there. So you've got more larval ticks kind of in your late summer. You've got your nymphal ticks in more traditional late spring, early summer, and you've got your adults in kind of late summer, maybe early fall. And so you've got 3 different life stages within 1 year that are occurring that we have to account for in the environment. So in terms of just studying this system, it's really quite complex. What's been done, so we've studied Lyme for a long time, and we've studied it in pretty specific environments. We studied it primarily in rural areas and in agricultural areas. These areas typically have a higher general plant and animal diversity, and they have either very high outdoor recreation, or the humans that are using these spaces are professionals that are out there. So wildlife biologists and foresters, and anybody who is in that, even farmers, are typically very familiar with Lyme. They've often had it. And so it's something for people who are using these spaces that they're more familiar with. It's also more widespread, it's less focused, although the human density aspect is much lower. And so it's been this kind of low-level chronic issue that we've not had a lot of good answers for. And so you see a lot of the science of how to address it has been focused there. Unfortunately, it's also more diluted. There's not simple places to focus on. Right now, my lab is looking at areas that Lyme and tick-borne diseases occur that are not these traditional places that we've taken a look at. So we're looking at areas that have higher human density, that might have good plant and animal diversity, but maybe not, have high levels of community gardening. And lo and behold, in a lot of these suburban and urban areas where there is green space, high levels of recreation. And so suddenly we're paying attention to areas that have not traditionally gotten Lyme research. And that is in these suburban and urban interfaces where we actually have very high incidence of Lyme. us to kind of the first project that I was not running the statistics for, but it launched all of the lab work that I've been doing since 2017. USDA ARS, a biologist there, Dr. Andrew Lee, started a project monitoring elementary schools in and around Prince George's County and Howard County. And several schools had come to USDA and were concerned because they had school children getting Lyme disease. And so they started monitoring, and they were quickly finding that the prevalence of Lyme disease in the mice and ticks around these elementary schools was so much higher than we would have ever expected. And that's what begged the question, what is this kind of perfect storm that we're creating in these areas we wouldn't have typically thought of as high Lyme incident areas? And so that keyed us into the idea of suburban areas with green space. And so we set up a 5-year project, and we were interested in covering kind of 2 big components. We wanted to look at deer as kind of the transporter of ticks and the supporter of general tick populations. We wanted to look at white-footed mice in particular, because they're kind of the heavy hitter bad guy for giving ticks Lyme disease, and then the ticks themselves. And coming from an applied scientist, I want to fix the problem. And so we looked at some potential IPM treatments. So we studied the system, and then we applied different sets of IPM treatments to try and figure out what might work. My objectives in general were looking at spatial data on the deer and spatial data on the mice, coupled with the prevalence of disease before and after any kind of treatment was applied. And so we had a huge field crew. We trapped deer for 2.5 years. We trapped mice for 3 years. We flagged for ticks and monitored vegetation. This is Dr. Andrew Lee, you see on the far right there. On the top left, this is how we trapped deer. Remember, these are pretty highly urban spaces, highly suburban spaces, almost urban. And so they're trapped under these drop nets, kind of looks like a circus tent that have been baited, and we're there on site and are able to drop the net and then restrain the deer and then anesthetize it. Mice were caught in huge arrays of normal Sherman box traps, so a small little box that's baited. In terms of how you actually deal with ticks, we've learned a lot from the rural areas. We've learned a lot about different border modifications, things like mulch or stone and having a pretty big swath. We've learned about cutting really scrubby vegetation. Interestingly, the black-legged ticks will climb out on vegetation and they wave their arms like waiting for something to walk by, as opposed to Lone Star that are really fast movers. We thought we were going to be able to use this product. It's a biofungicide that actually kills ticks. It's very difficult to work with. We have permethrin, which is kind of your classic treatment of tick killer. Then you have these 3 on the right. Demodex tubes and Select TS bait boxes actually use a compound similar to permethrin, but it's called fipronil. And then you have something called a 4-poster feeder on the top right. This essentially is a corn feeder, and you can see what looks like paint rollers standing on their ends on the ends. For the deer to access the corn, they have to stick their head in this kind of very small hole. And to do that, they have to rub up against this paint roller. You'll see some better pictures in a few slides. We know that deer carry most of their ticks on their neck and ear area. And so the concept of this is you're basically killing ticks by luring them in with a bait, which causes a tremendous number of problems. In island situations, those have been shown to be very effective. So we looked at these and we came up with these 3 options, and then we were really unable to get access to this biofungicide. And so it fell out. And so really it was left to 4 posters and these bait boxes. So let's take a quick look at those. Bait boxes. This is not like the typical ones you see where it's a rat or small mouse trap to kill them. This is just a standard bait feed with no chemical in it. In picture 1, you can see these mice are carrying multiple stages of ticks, primarily the nymphal and larval stages, although they carry adults as well. They come into the box and they feed on bait. And what you can see in this picture 3 is they actually walk through what they call it a wick of that fipronil. It's essentially like a car wash, kind of like the, the rags on the car wash that when you drive through. And so it's covering their body, killing the actual ticks that are on them, hoping to not only suppress the tick population, but also you're suppressing any ticks that might have fed on a small mammal that then are contracting Lyme disease. 4 posters, as you can see here, have been used successfully. In some states, though, they are considered illegal because it's considered baiting and congregating, and we see that happening more and more because of issues like chronic wasting disease in deer. So we don't want to congregate deer, we don't want to bait deer. Many places are already over carrying capacity. But in select situations where you can weigh the pros and cons, they might be the right choice. And so we included them because Howard County was already using them in some of their county parks. So we used them as one of our potential options. They did cause some interesting behavioral patterns that we saw in deer that we talk about in some of our publications. And you see this deer here defending the corn feeder. So I'm gonna try and show you this video, and I will rely on Luke or Taylor to let me know if it's not playing properly. It's a pretty short clip, but shows you some of us working up my master student and multiple of my technicians working up mice, because one aspect of this project was actually radio collaring mice. Some of my funniest stories come from that, and it's a good visual. So let's see if you can take a look at this video. Make this bigger.
So the long goal is.
We got our mother.
Over here. Oh, I got him. Yes. Okay. Yeah. We at the University of Maryland are working on this overarching project with USDA and Howard County, trying to address Lyme disease in these natural areas and in the interface of people's backyards. Lyme disease is a huge issue in Maryland and Howard County in particular. White-footed mice tend to maintain Lyme disease in the environment. They actually carry it in their bloodstream, and a tick that doesn't necessarily have Lyme can latch onto a mouse, contract Lyme, fall off, bite a human, and then give the human Lyme. There are a lot of mice that tend to carry ticks into backyards. So the idea is if there's a way to integrate treatment in and around people's backyards that reduce the number of ticks, then potentially we can reduce the chance of people getting Lyme.
All of the ticks' blood and tissue samples get tested by the CDC so we can get a report about who's infected and what ticks are infected. Besides getting the infection status on mice, we're collecting movement data and location data. Infection status of mice changes month to month, so we want to just track where the infection happens, the rate of infection per mouse, and if it lasts throughout the summer. They have a transmitter, caller, and I have a receiver. We have treatment out, and we want to see if the mice are moving at all to use those treatments. So hopefully we can get some information on if those beta treatments are the best way to treat mice for ticks.
An interesting thing about this particular project is that the mice, they're actually getting treated for ticks, so they're going to end up actually healthier in the long run. We're looking at the entire ecosystem to try and really understand the way we can best reduce the likelihood of humans coming in contact with ticks. What we hope to be able to do is to give a science-based management tool to people like Howard County Parks and Rec, homeowners, to be able to say, hey, here's something I can go out and do to make it safer in their backyards.
Okay, we're going to close out of that video and we're going to come back to the PowerPoint. Let me know if you don't see that now. We learned, so that project really is just concluding now, and we've learned a lot of lessons. Some of them very specific to IPM, some of them things that if we knew at the beginning, we probably would have done some things a little bit differently. In terms of the things that we learned from the mouse tracking and from trapping mice, is that there were kind of two ways to approach how might we address IPM that's directed at mice. So these kind of bait boxes and things like that. And there were two lessons that we walked away from. You could do a general population treatment, or you could try and create a barrier around places where humans were going to have very high amounts of use. If you were going to try and do a yard or barrier treatment, then to make sure that you were treating all the mice that had access to the human yard, that you would have to treat at least 40 meters into the forest. Beyond 40 meters or so, the mice that are there are never really going to come into a homeowner's yard. And then you would have to do multiple rows because of the way their home ranges are set up. So every 10 to 20 meters, preferably 10, you would have around 3 rows. Moving from the edge of their yard into the forest. And by that, you could create this barrier of protection of mice and ticks. At the same time, that doesn't do anything for the mice in the middle of any kind of green space, nor does it really do anything for mice that are living in and around the home. And so if you're going to try and do that from that standpoint of a population standpoint, using things like tick tubes or these bait boxes in a gridded system every 20 to 30 meters really throughout the park or around a person's home would be the complement to that. Perhaps you do both. We haven't tested those combinations because we needed the spatial data from the mice to figure out how much space do we need to cover in terms of protection. One thing we found out from mice that was surprising is that it's not just the mice that are carrying not only the ticks, but many of these diseases, zoonotic diseases, chipmunks, shrews, squirrels, many of the small mammals. And so in areas where we had less mice, we might have had a greater number of different species of small mammals that were playing that role. And so understanding the structure of the small mammal community and how that influenced what was happening in the Lyme and tick world, we didn't prepare for. We didn't have that information at hand. In terms of lessons from deer in suburban areas utilize neighborhoods as their home range. While they always maintain a core of their home range near some natural park area, especially females during the winter are completely living in neighborhoods. It's warmer, there's consistent food sources, there's less risk of predation. And so that was surprising to us that there was as much urban usage as there was, like around the neighborhood. I think upwards of 80 and 90 homes within their home range that they maintained these circuits of. And these 4-poster feeders are a nightmare. They are increasing the carrying capacity of deer in an environment that's already over its carrying capacity. They're very difficult to maintain, carrying big bags, 50-pound corn. The squirrels chew them and destroy them. If you're in areas with bears, it's a nightmare. And in general, we see them being used less and less. And so it's going to take a very specific situation to use those effectively. And so we feel like moving away from those is the way that most IPM is going to go for this. So it leads me to what the last topic I'm going to talk about today, and that is our new project that has started this year. Well, technically it started in 2021 with just camera work, but the real launch of it is this year. And we're taking all those lessons we learned from Howard and we're applying them in Montgomery to better capture this picture. What is the actual community in these areas? What is actually happening in terms of, you know, how many mice are there? How many squirrels? What are the prevalence of disease that they're carrying? How is that impacted by the habitat? Do really urban parks differ than truly suburban parks versus these rural areas? And so we walk away with, it's a much more complicated project, but we're gonna start to get at some of the things that are difficult to answer in these urban and suburban areas, but have just as much validity and concern as the issues in rural areas when it comes to Lyme. Now, you can see some of the big heavy hitters in terms of this type of science and ecology, and they've done most of their work looking at rural areas, and they're just now, or straight up IPM, and they're looking at ways to specifically treat in specific spots. Just in the last 2 or 3 years, have people started paying attention to this idea of really urban and suburban spaces and fragmentation and how that impacts it. And it's surprising because it's not necessarily obvious. So this is from a study that was published in, I think it was 2019 in Belgium. This is an area with endemic Lyme, meaning it's been there for a very long time and is well established. And you can see, depending on the connectivity and the community, you have areas that have really high prevalence with lots of a good community structure. You have areas with very few ticks, but they still have lots of prevalence. And then you have areas with really almost no wildlife and no prevalence. And so understanding what makes these different places a petri dish for Lyme or not, or really supportive of tick densities or not, is something that we haven't quite figured out yet. And fragmentation and the movement of animals is probably one of the big players, hence me being very interested. So we started this project in Montgomery County, and we assessed, all the potential parks across this urban to rural gradient. There's one park missing on here, Cabin John. And so we went in and we sat down with Montgomery and we came up with a plan. And we have deployed, in 2001, we deployed approximately 60 remote cameras randomly throughout 8 parks. And you can see that they move from very urban all the way to Little Bennett in areas that are much more rural. And so this was done in started in 2001. We're coming back in this year, and we've added 40 more cameras to these 2 blue parks down below. So that's Rock Creek, the bottom section of Rock Creek Stream Valley Park, as well as Cabin John Regional Park. We are going to concentrate very heavily on those 2 parks. In the other parks, we're looking at the density of all of the terrestrial mammal populations, excluding small mammals, and we're looking at ticks and vegetation. In these 2 urban parks, we're looking at not only the density of all terrestrial mammals, but also the density of small mammals, as well as ticks and vegetation. And so we're trying to capture the whole community. So we have these cameras set out. You can see Carson here on the left, and Grace, who you saw in the video, on the right. She's my PhD student. He's my master's. And Trinity is one of our undergrad techs who's working with us. We've set these cameras out everywhere. And I'm going to show you a sampling of what we've already seen. These are all coming from the urban parks, urban suburban parks, which is pretty fascinating. So raccoons, lots of wild turkey. We've started an offshoot of a whole nother project on wild turkey because of this data. You see major invasive Japanese stilt grass and what have you. We've seen the black bears way out in, this one is the one picture that's out in a rural Little Bennett. We were really pleasantly surprised to have captured some pictures of the bears, lots of foxes everywhere. Lots of deer, as you would imagine, feral cats, which is interesting, and we are recording those as well. Foxes right in people's backyards, basically right off a very small pocket park. More raccoons, foxes, and you can see how urbanized this is. In fact, that's a chair in the background. And of course, the deer are certainly an issue. We know that. And we have our share of the mesocarnivores as well, coyotes. So we're already collecting all of this data of what is where in these communities. And now we're going back in, and this is just starting this summer. We've got 6 pretty large trapping grids for small mammals. We expect to capture a swath of different types of small mammals. We will be collecting ticks from them and tissue. At this point, we're not collaring them, but we are estimating density and tagging them and collecting disease relevant data to start to build a better understanding of what the whole community looks like and how that relates to ticks. Only once we have that information can we come back and say, okay, how could we best apply tick tubes or bait boxes? Or counter to that, could we create a rapid assessment for county parks that they could go in, do a really quick camera survey and say, this is going to be a bad place because what we have or don't have, we should probably monitor for tick-borne diseases here. And so that's the goal of this project as we move into how to better address not only tick issues and education issues in rural areas, but these areas that kind of snuck up on us, these suburban and urban areas where people are actually contracting more ticks. We've documented that. Dr. Scott Williams from the Connecticut Experiment Station published data showing that people are most getting ticks in their yard, gardening, mowing the lawn, walking along the edge of their properties, just as much, if not more, than where we see that what we would have thought as traditional contraction of ticks. So when we're trapping, we're going to be looking for your kind of traditional mice species that we might have, mostly white-footed shrews. This is, I think, one of their Snapchat accounts of my students. So they like to do these kind of fun things. And then flying squirrel is also another that will likely be caught in traps along with the other squirrel species that we have. Okay, so I'm going to end there. Hopefully I've answered a bunch of questions. If not, I'm really excited to follow up in anything you're interested in. Would acknowledge ARS. One point of difficulty for these projects are no one wants to claim responsibility for Lyme that's not directly agriculturally related. And USDA ARS has stepped up to the plate in terms of let's take a look in these suburban areas and see if we can figure something out in these urban areas. Because who's responsible for ticks on private property? Counties often don't have the funds to do big projects like this. And so it's been a big conglomerate of support for this project, Montgomery County and Howard County, of course, my department, and ARS thus far. With that, I'm going to stop sharing, and I'm happy to take any questions that you might have about the project or about Lyme and our future work.
Thank you very much, Dr. Mullinax. Great to see a good overview of your work. I learned a little bit more. And I know I have some questions, but I'm going to pause and see if our audience has some. Anything coming through in the chat? I'll give them a few seconds. Maybe I can ask a question or two, and then hopefully we'll get some coming in. I'm curious, you mentioned all the different small mammals. Are you finding different, are you able to test that the animals are infected? Do all of the different small mammals get infected with Lyme and then pass it on to ticks? And do you find different rates of infection between different small mammals?
Yeah, so that's a great question. We did not anticipate the number of different small mammals that would not only be carrying ticks, but that would be carrying Borrelia positive ticks. For all the small mammals in Howard, we collected ear tissue. So it's a little tiny hole punch. Imagine getting your ear pierced, but taking a little piece of that tissue. From that tissue, they can test for a suite of different tick-borne diseases, and we did that. And we're surprised at the prevalence in shrews in particular. And there is, we knew white-footed mice would be a huge culprit, but actually every species that we caught, we had some percentage that was carrying a carrier of these. So can I speak to the percentage of the population? No, because we weren't doing it for density for the other animals. And in fact, Howard County wasn't set up to estimate density of them at all. It was sheerly like for prevalence, like is it out there? And how much do we see across small mammals? And so the Montgomery County study is designed specifically to assess density. So I hope to have more clear answers on that. But we do know that these other species carry it. All of the small mammal species seem to carry it. Terrestrial mammal species seem to carry it. And we're interested in shrews in particular.
Wow, I would not have thought about shrews. All right, we have a question coming in, I believe, from Art Smith here. At one time, there was a vaccine for Lyme disease, but it is no longer available. Could you maybe elaborate on that?
Yeah, sure. You know, it's so funny because I asked my veterinarian about it. I was like, could you just give it to me? Like, is that legal? You know, and I have to be concerned with all of the student workers I have. I have a pretty big crew. Like this summer alone, I think we'll have 15 people out in the field in places we know that it's really heavy Lyme. The vaccine was very ineffective in humans, and there was a pretty significant immune response to it. And so, you know, that's pretty normal for vaccines. You get a vaccine, your body like builds antibodies and has an immune response so that in the future, if you're exposed, it's, you know, it's ready. People don't like getting sick after vaccines, even if it's a normal sickness. So that was one problem. And the second problem was it had less than 50% efficacy. And given those two pieces, it was not supported by the drug industry, like they weren't making money off of it. And so it was discontinued. That's my Reader's Digest version of what I know. For dogs, however, they do the booster, and, you know, I guess we kind of ignore if our dogs feel bad, like we don't necessarily register that as much, but 50% upwards to 70% effectiveness, it's worth it for them, because it's, you know, dogs can't tell us when they feel bad. And so it seems to coincide well with them going ahead and continuing to do it. And it's essentially the same vaccine. That's the best I can answer, Art. Hope that helps.
Cool. Keep the questions coming in, audience. I've got a few more. I'm going to throw another one out there for you. This is kind of a fun one. Oh, good. We just got Tanya Shui just wrote, can a homeowner set up cameras like these to get an idea as to what animal they're visiting their yard?
Oh, sure. Yeah. In fact, we have cameras all over that are not ours. So they're just remote cameras, wildlife cameras. You can buy a pretty good camera for less than $100. Typically it uses an SD card that you can then download into your phone, or you can get a Bluetooth SD card that just goes straight to your phone. Now they have really fancy ones if you want to spend a little bit more money, that the camera sends it straight to your phone or computer. Lots of people do this. Lots of hunting population people do this to see what's out there, and lots of just general interested homeowners do it. And I've set them up with my kids in our backyard for their own entertainment and to see what's traveling through our space. I highly encourage it. I think it's really educational. So many of the wildlife species we love and are interested in, they only walk through really early morning or late at night. These cameras typically have an infrared light on them so they can take nighttime pictures. You can get them from Amazon, from Bass Pro Shop, anywhere, a kind of outdoor store like that will sell them.
Cool. I invested in some of the cheapies from Walmart for $35, and they worked pretty well. I have 3 out of 10 that are still operational 2 years later. So
that's pretty good.
You have failure rates that increase, I think, the less you spend. So,
yes.
And I'll say we're currently using a bunch of Browning cameras. We've had good success with those right now. I'll just put that out there, not endorsing it, but
yeah, Browning and Remington are two big brands. We use Bali, which we really love. There's a couple very specific science brands that are kind of the cream of the crop, but you pay a lot of money for it. So
yeah. $400 cameras are a little bit high for me, but yeah, you can get those at Reconics, and they're highly reliable and highest quality you can get. I got another question. How do you trap flying squirrels? Can you catch them on the ground with those Sherman traps, or do you have to have?
So they straight up go in our normal mouse traps. It's not, there are other ways to trap them in tree traps, and it's essentially like a trap attached to a tree. But these traps are baited with essentially peanut butter and oats, and they're attracted to that like any other squirrel. And they crawl right in. And so they smell it and they investigate it. So people think flying squirrels always flying, but they're not. They have plenty of, terrestrial typical movements. Yeah, and so we are legit just trapping them in our normal traps.
Cool, cool. I'm curious also, I know you've probably got a lot, this is a highly complex system, and I can only imagine trying to untangle it all. Do you have an intuition as to kind of the most effective method that you think is worth pursuing here? Or is this something we should live with, or do you use it only in high risk areas?
Yeah.
I'm curious, a high level thoughts on that.
Yeah, you know, we have really acute issues like COVID. I think COVID is the same kind of zoonotic disease. I think Lyme disease in particular is a chronic disease that there is no one answer for. So I am a huge proponent of IPM measures, so integrated pest management measures. That means you do multiple things and try and hit it from many different pieces. I think education is one huge issue, especially in the suburban and urban areas. Montgomery County and Howard County have consistently tried to do really good deer management. Their deer population is over its carrying capacity, and that is directly related to maintaining high tick populations, regardless of disease, but the number of ticks is really high. And when you have really high tick populations, you're going to end up having a greater chance of having outbreaks of Lyme disease in those ticks. So I think good deer management is number one. I think that the tick tube / bait box is probably the best IPM metric we have right now. The problem is that it's not been analyzed enough in these kinds of situations to figure out how to best implement it to make it effective. Given the tick life cycle, we already know it's going to be a multi-year. So it's like if you identify a hot spot, and this has happened in Howard County, I walk down a street and 8 out of 10 homes, these homeowners have had Lyme disease. It's insane. You identify spots like that and you say, okay, we're going to go in and we're going to do triage in this area for 3 years. This is what we're doing. And we're consistent with it. We're covering the right seasons and we're knocking the ticks way back. Mice don't live more than 2 years. So if you knock back tick populations and you give the mice time to cycle through that they're not contracting it from other ticks, then you can suppress it long enough to get you to a point where you're much less of a prevalence level, potentially reduce it to almost nil. The problem is that it takes this attention and effort and funding for this period of time, which, you know, it's difficult to do. We're all broke and we have short attention spans. And so I think it's those pieces, the education piece, the deer piece, and then figuring out how to apply the specific small mammal piece. I do think there's we're going to find communities that don't have it, and we're going to wonder why. And I think answering the question of why, communities that have more predators, we might see less of an issue because they're putting a check on the small mammal population. Areas that are completely cut off, we could probably treat better. We also know turkeys are actually playing a role in this tick cycle somehow because they carry so many ticks. I think you and I have talked about that, Luke. And so we're pairing the tick study with ticks in Martha's Vineyard. That is a complete island with massive turkey problem and massive tick and Lyme problem. So we're figuring it out. But it's going to take a little bit because it is incredibly complicated.
Can a homeowner make their own tick tubes, and how effective are they?
Yeah. So my sample size of this is pretty low, but I did it. So I live up against a huge forest right off some USDA property right in Greenbelt, Maryland, so urban, suburban. And we had horrendous ticks. And I mean, we were sampling in that forest behind my house. So I did my own little experiment and I mulched, and then I actually bought the Damidex tubes from like Home Depot or Lowe's, but you can make them. All it is a toilet paper roll stuffed with cotton with permethrin. You can buy permethrin from the big box stores. Depending on the concentration, you're only going to be able to buy what's commercially available. We typically use a little bit stronger stuff, but it's the same. You can do it yourself. And it's the same thing that's in bug spray, like the really hardcore bug spray. So You soak the balls in it. You don't want it to get it on your body or skin or anything. You put it in the tubes. If it were me, I would probably get some like black duct tape to protect the toilet paper tube because it'll just degrade so much. What happens in this case is that the mice go into the tube and they take the cotton back to build nests. And so they're taking, it's a little different than the bait box. They're taking the cotton that's been soaked in the permethrin into their nest. So they're treating themselves and their offspring by using that. The key with those is you just spread them around the perimeter of your yard, and you need to do it like twice a year, kind of spring and late summer, and you need to do it consistently over a couple years. I did it in my yard, and my kids stopped bringing ticks in the house. My dog stopped bringing ticks in the house. And so I've continued to do it for the last 2 years, and will continue to do that moving forward. The other thing is it has much less impact than things like that fungal spray that we were potentially considering using, because you're not really impacting any other species. You're not spraying a chemical that's going to kill a bunch of other bugs. You know, your impact of that is the footprint is very small. And so I think that it's a really good thing for a homeowner to use if you know you have a tick issue.
Cool, that is awesome to know. I did not know how that you could have such success. Are you finding that other, I guess, are the mice the main culprit? Do you think that's kind of the main, like, are shrews also picking up some of this stuff? Do foxes carry these things? And is it just like if you can hit the mice, you can kind of knock out the base of the pyramid kind of thing?
Yeah. So something that people don't realize is how many mice there are. And, you know, the white-footed mice are the main culprit. We feel very confident in that. And when you walk from your house to the back of your yard, you've passed a couple mice in the structures and around your home, in the wood pile. When you walk into the woods, you are surrounded by them in the trees, on the ground. I had no idea they even went in trees. I'm a wildlife biologist, and this was something I didn't know. They live in them. They're in them all the time. And so I think people just, because you don't see them, they significantly underestimate the number that are out there. So hundreds within a square hectare. So we feel very confident that they are the main culprit. In systems that have less mice, we're interested to see what's carrying it. We know shrews carry it, but it's hard to get true densities of them. They die really quickly. So we have to trap them very carefully because their metabolism is so high. The complement to that is systems with lots of mice with no Lyme. We're very interested in what's going on here that's different. Is it the soil? Is it the vegetation? Is it the fragmentation? And so, you know, we're just not really sure exactly the different mechanisms, because you can have these different components that reflect very different systems if you don't understand the driver. But we feel very confident that the white-footed mice are the big driver.
You mentioned their spatial movement and that they will not move more than, I think, 40 meters is a safe space. So it sounds like you trapped them and put little collars on them and saw their home ranges. I'm curious if you could just elaborate on what did that look like?
I have one here. I have a collar here, and you probably saw it in the video. So this is a little tiny mouse collar, and it's a traditional VHF collar with a little antenna. And we hope to do it again, but we did that in Howard. You probably saw that in the video, them putting the collar on. And we had a crew that went out. The problem with the small collars like that is you have to manually track them. So it takes 3 people in the woods, and mice are active at night. So they started at dusk and then tracked until they had no movement for 40 minutes. So they usually by like midnight, 1, 2 in the morning. And we were able to estimate home ranges and movement rates. And we were able to see compression of home ranges. And one thing people don't think about are mice are incredibly territorial. And so, especially female mice. And so they very much defend their territory. They aren't nice to each other. And so because of that territoriality, depending on the resources that are available, they can compress or they can expand, but they maintain these territories. And what that does is it creates a buffer. So you can hit enough territories, then you can create a buffer to prevent these other mice from being able to access people's yards. And so that was kind of the idea behind wanting to understand their home ranges and how they moved within a small space, a small population of mice. Now, we did have mice that were right on the edge, and I had my graduate student call me at like 11 o'clock at night, and these mice have those little silver ear tags, and they have the radio collar, and the mouse had gone into a person's home, and she tracked it into the house, and she called me, and she's like, what if they see it and it's running around with a radio collar? And so we had a lot of mice end up going in and out of people's barns and garages and decks and then homes from that edge of field. And so it's just something you can't prevent for the most part. You can typically prevent them from coming into the house, but the out to exterior and outbuildings, it's almost impossible. And so that was very enlightening for us in terms of, okay, we really need to make sure the mice on the edge here are getting treated the way they need to be treated. But the interior forest mice, they stay there. They don't come into people's homes. And so that was what gave us the idea of the barrier.
Very neat. Cool. We have 1 more minute. And I don't see any other questions, and I have lots of questions. I think I've seen lizards with little ticks on their faces, and I don't know if snakes I've never seen a tick on a snake, but I wonder, are reptiles, are they hosts to this? Do they get the same ticks? Are they separate types of ticks? What's the reptile angle on this?
Okay, you're welcome, Peggy. Thanks for that note. So I glanced over this slide, but on the slide of that really kind of messy ecosystem, if I had brought your attention to it, which I didn't because we're not doing reptiles right now, but reptiles are considered a diluter of Lyme disease. And so as opposed to the small mammals, which foster Lyme, reptiles dilute Lyme. They are considered an end host. So ticks that feed on snakes or lizards, we know lizards especially, they can give the lizard Lyme, but these lizards seem to be very robust to just like they deal with it, and they don't tend to give other ticks the Borrelia. And so they're considered a dilution host. And so that's fascinating to us. And it's an area that needs to be researched more, especially in the southern regions. And as these tick-borne diseases creep south, we think, like ecologically, that might be one of the reasons we've never seen it push so far south, is because as you move further south, you see an increase in these reptile populations. And so ticks are likely feeding on them much more. And maybe that's one of the ways to keep it diluted. So we aren't covering reptiles in this particular section of the study, but we hope to in the future.
Cool.
Yeah.
Cool. Neat stuff. Well, we got some nice comments. Alan mentioned he enjoyed your presentation. Yep. Thanks, Alan. And thank you, everybody, for joining us. This will be recorded, and it'll go up on YouTube, and we'll have many more people join and learn about your research there, too. I want to say thanks to everybody for joining, and I think with that, we'll say one last round of applause for Dr. Mullinax coming and sharing.
Thank you. It was my pleasure. I really appreciate it.
A Zoom applause occurring there, and yeah, it's good to hear from you, and look forward to being in touch.
Okay. All right. Wonderful. Thank you so much.
Bye-bye.